Hi students, welcome back to Chapter 5, Energy and Enzymes. And in the previous lecture, in Part 1, we learned a lot about the different types of energy, potential energy versus kinetic energy. And we also learned about exergonic and endergonic reactions and how those two reactions are coupled together. Remember that exergonic reactions release energy and endergonic reactions require energy. So recall that the form of energy that cells are able to use is a molecule called ATP, adenosine triphosphate. And that's the molecule that powers nearly all forms of work that occur inside of a cell. And if you look at what you see here, this is a drawing or a representation of ATP, where you have in green here, this is something called adenine, and then you have a sugar, a monosaccharide called ribose, with 3 phosphate groups. So you've got 1, 2, and 3. The way that ATP is able to provide energy for a cell to do work is during chemical reactions that require ATP, one of these phosphate groups comes apart, or that bond between the second and third phosphate is broken, and this releases energy. And so one of the reactions we should learn about is what's called what we've learned about is hydrolysis. So when hydrolysis occurs, when hydrolysis of ATP occurs, it releases that phosphate group. And remember, hydrolysis requires a water. So a water molecule is going to break that bond between the second and third phosphate, and the result is a release of energy. We've learned about hydrolysis before in the past. So if we think about the types of work that have to occur inside of a cell, there's going to be what we call chemical work, and that's going to drive chemical reactions inside of the cell. So if you have some sort of reactant here, and you'd like to put, or 2 reactants rather, and you'd like to put those 2 reactants together, phosphate group pops off, and then you get this new molecule that's formed. Mechanical work is the work that causes movement inside of a cell. So your muscles, when they contract, the individual proteins inside of a muscle interact with one another to cause that movement. ATP is required for work there. And then also, a few lectures ago, we learned about transport of molecules across the cell membrane. And remember that some of the transport that occurs across the membrane does not require energy. Remember, that's called passive transport. Whereas active transport is going to require energy. And ATP is going to be the form of energy that drives that transport work as well. So we lost a little bit of formatting here, but I think we still can still see what's going on. So when we were learning about energy coupling, those exergonic reactions driving endergonic reactions, more specifically, when we're talking about ATP, there are two reactions here, one called phosphorylation and the other called hydrolysis. ATP is a molecule that's constantly being regenerated. In an exergonic reaction, one of those phosphate bonds is broken and energy is released. And in endergonic reactions, a phosphate is put back on. So if we think about phosphorylation, that begins with a molecule of ADP. That means adenosine diphosphate, meaning there are just 2 phosphates because one of those phosphates has popped off. That's what ADP is. We would like to pop a phosphate back on, that ADP, adenosine diphosphate, to make adenosine triphosphate. And that's a reaction that requires energy. And the energy comes from those exergonic reactions. So this is the reaction called phosphorylation. So adding a phosphate to ADP is called phosphorylation, and that makes ATP. And then when ATP is hydrolyzed, when a phosphate group pops off, energy is released. So those two reactions are coupled, phosphorylation and hydrolysis. Phosphorylation adds a phosphate, hydrolysis takes away a phosphate or breaks that bond, and energy is released. So that's some of the reactions that occur to, or how cells use ATP for the various processes that occur inside of a cell. Now what we're going to learn about are enzymes. And enzymes are proteins. All enzymes, or virtually all enzymes, are proteins. And enzymes are going to be these molecules that help reactions occur. And the important thing to remember about enzymes is that they don't make reactions occur. Chemical reactions inside of a system or inside of a cell would naturally occur if given enough time. But what enzymes do is they speed up those reactions so that they occur in a time scale that's relevant to the cell. So we're going to learn now about how enzymes drive cellular reactions. So let's just look at some of the details here. Enzymes are referred to as biological catalysts or helpers or molecules that are going to help speed up reactions. And the way that they work is they lower what we call the energy of activation. They reduce the amount of energy for a reaction to begin. So they increase the rate of a reaction, or they make those reactions occur in a timely manner, but they're not consumed by the reaction. They are not consumed. They're always recycled so that they can continue to do their job over and over and over again. Oftentimes, enzymes are named for the reactions that they cause or they catalyze, or they're named for the substrates that they bind to. So just as an example, lactase is an enzyme that breaks down lactose. Lactose would be the sugar that we find in many dairy products like milk. So if you're lactose intolerant, that means that you are no longer making lactase, an enzyme that breaks down lactose, and then that lactose causes digestive upset. Alternatively, there is an enzyme called alcohol dehydrogenase that breaks down alcohol. And so if you're lacking in alcohol dehydrogenase, even if you consume a little bit of alcohol, you may see much greater effects than somebody who does produce that alcohol dehydrogenase. That enzyme is required to break down alcohol in the liver. So let's look at the graphic representation of how enzymes work. If we were to start out, let's just imagine this is the left side of the arrow here. Here's the right side of the arrow. We'll just draw in an arrow so we recognize that there's a chemical reaction occurring. If we start with our reactants, you see that this is, for instance, what they're showing you here is a disaccharide, 2 sugars. And what the reaction that should be occurring is we want to break that bond. We want to break up that disaccharide into 2 monosaccharides so that what we get is these products here, 2 monosaccharides. The energy required for that chemical reaction to move forward is represented by this bar right here. So that's the energy input that is required for that reaction to occur. And over on the left here, this is the amount of energy that would be required without the enzyme. But if we look over on the right, we see that same energy bar right here has been lowered. So with an enzyme, the amount of energy required for that reaction to occur is much lower. So the bond is broken and you get these products with the enzyme. Now, more about enzymes is that they're really selective in the reaction that they catalyze or cause to speed up. And the shape of an enzyme is incredibly important. Remember, all enzymes are proteins. And remember what we've been learning about structure and function, that the structure of a protein is going to dictate its function. And so specific reactants are only going to be acted on by specific enzymes. So we call this enzyme specificity. Certain enzymes will only catalyze reactions for certain reactants. So what the molecule that an enzyme works on is called the substrate, and they fit together like a lock and key mechanism or two puzzle pieces. The substrate, the molecule that the enzyme is going to act on, fits into a region or a location on the enzyme called the active site. And I'll show you a picture of this in a second. So there's an area on the substrate and an area on the active site of the enzyme. They fit together like two puzzle pieces, and then their reaction moves forwards. So we say the enzymes are specific because their active site only fits specific substrate molecules, two puzzle pieces that only fit together with one another. So earlier on in the semester, we learned about how proteins are made of amino acids, so chains of amino acids, right? And then those chains of amino acids get really long, and then those chains start folding up into sheets, and then into barrels, and then into more complex structures with multiple barrels. So enzymes usually are only going to be functional when they have this really complex shape. But if this enzyme loses that complex shape, it can no longer catalyze its reaction, or it no longer fits with the substrate. So if we look at how this occurs, here's an enzyme called sucrase, and it's an enzyme that breaks down a sugar called sucrose. And if we look at the enzyme shape itself, you see that here's the active site and has a very specific shape. So when sucrase comes in contact with sucrose, what will happen is the sucrose then fits right in that active site of the enzyme, and that active site on the enzyme only fits sucrose. The active site changes shape a little bit. The reaction moves forward. You can see here that the bond is broken between those two molecules, and therefore that substrate, that sucrose, is converted to products. And what's required for that reaction to occur is water. And so this is a hydrolysis reaction. Remember hydrolysis, the lys means to break, and so this is breaking with water. So we've broken that bond using a molecule, and then what you get are the products. So you've got a molecule of glucose and a molecule of fructose. Those are released, and then the enzyme goes back to its original shape. So the enzyme is recycled, and then it can go ahead and bind with another molecule of sucrose, and the whole process then just kind of continues. A blank slide here. Just want to show you this because this is something that we'll talk about. So we know that when we put fuel in our vehicles and we combust that fuel, that one of the products that comes out is carbon monoxide. We just say that that's CO. But also there's some carbon dioxide from incomplete combustion of the fuel as well. So these are some of the products that are a result of breaking up the molecules in gasoline, right? So we've got carbon monoxide and carbon dioxide that get released into the atmosphere. Well, what goes up must come down. So that CO2 that gets released into the atmosphere eventually falls down into our lakes, our rivers, and our oceans. And so I'll talk about the oceans specifically. We've got CO2, and when that CO2 makes its way into the ocean, it combines with the water in the ocean and creates that carbonic acid. And as a result, we get, over long periods of time, we get the pH of our oceans decreasing. Our oceans would really like to be somewhere around 8.2, so somewhere just above neutral. But what's happening is, as we put more CO2 into the ocean, we're continuing to decrease the pH of the ocean. And what happens is that decrease in pH starts to affect the enzymes that all of the creatures living in the ocean require for their cellular activities. Remember that temperature and pH are the two key factors that are going to affect enzymes. And if an enzyme's structure, or that protein structure, is changed, that enzyme no longer works or no longer works properly. So as we have a decrease in pH of our oceans, that's starting to affect the enzymes of all the organisms that live in the ocean. And this is a process that we refer to as, you may have heard of it, ocean acidification. And this is one of the effects of climate change. Climate change has in the past been referred to as global warming. And that's no longer the appropriate term to use for all of these consequences as a result of putting more CO2 in the atmosphere. So we don't really use global warming as a term anymore, although global warming does occur. It's just one of the symptoms of climate change. So we've got the pH of the oceans decreasing, we've got temperature of the atmosphere and of our oceans increasing. So if you think about what that does to enzymes, you can see why these are very concerning issues for organisms living in the water. At the end of this lecture, and I won't post the link here, but I'm going to post the link on Canvas for you guys to watch. And it's a video that was done by a group that I used to work with in Indonesia, and it talks about the effects of the amount of CO2 in the atmosphere on the oceans, more specifically coral reefs. And coral reefs are living structures. They look like stone, but they've got a little tiny animal on the inside that builds the hard outer skeleton. So coral reefs are living. And coral reefs are the kind of the canaries in the coal mine, so to speak, for climate change. Because as CO2 increases in the water and pH decreases, this has a negative effect on coral reefs. They can no longer build their outer protective skeletons as they would normally, and the existing skeletons start to slowly dissolve. The skeletons become weaker. And this is causing coral reefs to die at an alarming rate. And it's very concerning. And it's something that I will continue to come back to as we move along through the course. The video that I show, or I'm going to ask you to watch, is only 12 minutes long, but it is teaches you a little bit about the effects of high concentrations of CO2 in the atmosphere. So I'll have you guys watch that for a little bit of extra credit. Anyway, that's a side note, but it's just one way to relate pH and temperature to the real world, what we see happening around us with climate change. So in this week's lab, I believe it's this week's lab, you guys are going to be learning about enzymes and some of the things that affect enzymes. Enzymes usually have what we call optimal conditions, or the best conditions under which they're going to convert the most reactants to the most products, the conditions under which they work best. And temperature is the biggest one. And temperature, what it will do is it can, certain enzymes have a specific temperature range that they would like to work in. And so if temperatures are too high for an enzyme, it can ruin the shape of the enzyme. It ruins the shape of the protein. And remember that the protein's shape is what allows it to carry out its function. So if the protein's shape is changed, the enzyme won't function properly. So if we think about in humans, for example, most human enzymes work best at a temperature between 35 and 40 degrees. Human body temperature in Celsius is right there around 37. So imagine how a fever would affect enzymes. A high fever for a sustained period of time is going to start affecting all the enzymes in your cells, and therefore cells can't carry out their daily activities, and potentially then cells begin to die, tissues begin to die, organs then start to fail, etc. So this is why we really strive to control a high fever. Another thing that affects enzyme activity is pH. I remember when we were learning about pH, that pH is the measure of hydrogen ions in a solution, where we measure it on a scale from 0 to 14, 7 being neutral. Now, most enzymes really like to be right around neutral, somewhere around 7. But there are exceptions. Enzymes in the stomach, where the stomach, for instance, has a pH of like 2. Enzymes in the stomach can work at a pH of 2. They're very specific. But just in general, enzymes really like to be somewhere around neutral. And so let me just give you an example here. All right, up on the top, let me erase a couple of things here. All right, I haven't mentioned this yet, but this is true just kind of in general with carbon dioxide. When carbon dioxide is mixed with water, what you get is something called carbonic acid. So if you were to hold your breath, CO2 builds up in your blood, which is a watery solution. Then you get carbonic acid building up in your blood, and that drops the pH of your blood. Your blood would really like to stay at a pH of somewhere around 7.2. But when you hold your breath and CO2 builds up in your blood, which is water-based, you start creating carbonic acid, which drops the pH below 7.2. And you've got receptors in your brain that can detect the pH of your blood, and they start sending signals to the brain saying, hey, there's too much CO2 in the blood, you gotta breathe, you gotta exhale. And that's what you feel when you're holding your breath. So if you go swimming or you just hold your breath for whatever reason, you know that after some period of time that you're going to feel this overwhelming urge to breathe. And that's your brain saying the pH of our blood is too low because you're not exhaling that CO2. We're going to have some issues here. You need to get rid of that CO2 to bring that pH back up. And this is also true, this type of reaction, just really basically. Okay, so we know that enzymes have what we call specificity, that they're only going to catalyze specific chemical reactions inside of the cell. Some enzymes, though, require molecules that are not proteins to help carry out the reaction, and these are called cofactors. So what cofactors do is they'll bind to the active site on that enzyme and reshape the active site so that it fits the substrate. A lot of times these are metals, so they're inorganic, they're not carbon-based, so they can be things like zinc, iron, copper are just a couple of examples. If it's an organic molecule that's acting as a cofactor, we call it a coenzyme. So vitamins are often referred to as coenzymes. So a lot of cofactors are inorganic, but if it's organic, we call it a coenzyme. So these are helpers that are going to help a reaction occur. Additional molecules help a reaction occur. So in this picture, what you see is the enzyme, the coenzyme, which would be something organic like a vitamin. And you see the coenzyme on the right of the arrow. You see that coenzyme moving into that active site, changing the active site. So now that active site fits the substrate that, or the molecule that the enzyme would like to break down or build. In some cases, there are what we call enzyme inhibitors. We don't always want enzymes to continue to catalyze chemical reactions indefinitely. There has to be a set of breaks, right? So there has to be some point where the enzyme can no longer do its job, at least for a period of time. And so these are what we call enzyme inhibitors. So an enzyme inhibitor might work like this. If this is the normal active site, you see the substrate's going to fit right in that active site. You see how inhibitors can work. They get in there, kind of get in the way of that in that active site, so that that substrate can temporarily not bind. So this will stop a reaction in its tracks. So this is called a competitive inhibitor, where it competes for the active site itself. A non-competitive inhibitor will bind somewhere else on the enzyme, and that binding elsewhere on the enzyme also causes a change in the active site on the enzyme, so the substrate can no longer bind. Okay? Remember, we don't want chemical reactions, all chemical reactions, to continue indefinitely. There's got to be some breaks. So the reason we have these inhibitors, as I said, was there has to be a set of breaks where we can slow down a reaction or stop a reaction temporarily. So these enzyme inhibitors are really important in regulating a cell's metabolism, or the rate at which those chemical reactions occur. In some reactions, the product, the thing that's made, may act as an inhibitor. So if you get enough of a product created from a chemical reaction, that product itself can stop the reaction. So once that product gets to a high enough concentration, the product acts as the inhibitor. So let's say we've got this, it's a little bit more complex of a reaction here, where we've got reaction number 1 that converts A to B with enzyme number 1. And then we've got reaction 2 that converts product B to product C. And then we've got reaction 3 that converts product C to product D. And product D, for instance, can act as an inhibitor in this first reaction. So that would prevent all of these other reactions from occurring when we've got enough of this product right here. And this is something that we call feedback inhibition. So the cell is really giving feedback to itself in the form of this product. When this product is in high enough concentration, that provides feedback to stop this multi-reaction process, at least temporarily. And then when the concentration of that product goes down again, this reaction continues. So you can see how this would regulate a cell's metabolism. One example here that you might be able to relate to is blood pressure. The amount of pressure put on, created by your heart and put on your vessels would really like to stay kind of right around, or swing right around, what we call a set point. So we can just, it doesn't matter the number here, but we'd like to just have it right here along this dotted line. We don't want to have huge swings in blood pressure. We don't want blood pressure to go too high, and we don't want blood pressure to go too low. We want it to stay stable. So what happens in the body is that signals, when the blood pressure gets too high, signals, enzymes in the blood cause a decrease in heart rate. And that also causes blood vessels to vasodilate, and that means they dilate, they get larger. And when those blood vessels get larger, blood pressure goes down. But if that blood pressure gets too low, like you see right here below that set point, other enzymes signal the heart rate to increase and cause the blood vessels to constrict, to get narrower in diameter. So those enzymes involved in making blood vessels wider and making blood vessels narrower help to regulate our blood pressure right around this set point, helping to maintain that very important state, homeostasis. Okay, so there are a couple of slides here that just kind of remind you of some of the things you should know. And I won't read these to you, but you can go through them in the video, but you can also find these slides in the folder where you have access to all of my PowerPoints so that you can read these. So there's this last one here. These are kind of your checkpoints, your knowledge checkpoints, where you, if you can explain, describe, or define these things, then you're doing a pretty good job in your studies. Okay, that's all I have. This is the end of the Chapter 5 lecture. See you next time.